Nice to be here. Thank you, John, for having me here. It's a wonderful meeting to see everybody working on Drought. I'm really honored to be here talking to two other Johns. People I've heard about for many years, John Pasiora and John Boyer, who have sort of trained my mind in what drought biology is all about. As John said, I started as a breeder. My only breeding experiment was in wheat and doing mutation breeding. And after that, I decided that actually mutants were much more fun. So I started working with mutants, and those were mutants of transposons, because in those days, transposons were the only real models of gene regulation. And so what I'm trying to get now to you today is some sort of a regulatory network in which we can understand a lot of drought biology. So I'll be talking essentially about drought stress response and resistance, looking at it in terms of a very complex biological process, because there's a lot of interplays, and you all know there are a lot of definitions of drought. Everybody has got their own pet ways to address it. When I started in drought research, the first thing I did was do drought really in the soil to begin with. Started with a Rhabdopsis, was a good system. We got a number of mutants. And then I moved to rice and maize, especially since rice became a very good model to be studying it at the genome level. So I'll talk about drought stress response at the transcriptome level, because that's one of the easy readout systems. Of course, transcriptome is various levels now of regulation. It can be at both at the processed RNA, unprocessed RNA, small RNA, et cetera. So even that is a very complex system. And to understand this all, we look at it in terms of networks, because networks gives you a logic, essentially, of why these genes act together and how they interplay together. And to probe these networks or probe these sort of models of regulatory behavior in terms of drought, we use stress regulatory genes. So mutants of them or certain perturbations in them, and therefore look at what the downstream effects are. The two aspects I've been looking at are drought stress resistance in its broader context and water use efficiency. And what I'll be talking about today is also certain models which we have in genes which we have identified, which address essential biological processes, many of which you know, which are involved essentially in drought. So the basis essentially is when we study genes and looking at these genes by themselves, they give us an idea, but it's not enough. So what we like to look at is how genes interact with each other in modules. Let's say something is switched on or off, and how they all interplay and work together. So these modules can be biochemical pathways or certain biological processes at a lower level. And then all that could work in terms of a network. So a network would be something like in terms of a network which essentially is flexible, and which is what you see finally when you give a plant some drought stress. So to begin with, we started with rice and maize and look at transcriptome studies. Studies had shown that, of course, the most sensitive stages of drought in cereals was the reproductive stages. So we looked at the panicle of rice and ovaries of maize. So maize ovaries directly a day or two after pollination, and panicle elongation stage. And we wanted to find out what were the features essentially which affected there in terms of drought response. And our readout was in terms of transcriptome expression patterns of all genes. For rice, we looked at Affymetrix arrays and also did some small RNA-seq. I won't talk about that today, but we did RNA-seq for maize because, the maize genome is well known, but many of the genes essentially are probably not well annotated enough to find out. So we thought we'd have some more gene discovery, and I'll show you some parts of it. Now, why are we interested essentially in these reproductive stages? One is because persons like John Boyer had shown that essentially during drought, both in rice, maize, and wheat for other workers, we have very sensitive stages in which the sucrose or hexose levels go down, mainly due to reduction in the level of cell wall invertases. which essentially could lead then to cell death responses and no starch in pollen and grain or kernel number reduction. So this is a very interesting stage to study. The most interesting thing I found out there was that the cell wall invertases are not only at the regulatory level of the protein, but they're also transcriptionally regulated. So once they're transcription regulated, it will be interesting, therefore, to find out what is the transcription pathway involved in this shutdown, and can we study that in terms of regulatory processes which would keep that up. So that was the critical first step of starting. In Maize, we looked at RNA-seq. We had 8 libraries in both reproductive tissue and vegetative tissue. We had 0.3 billion Illumina reads. The most interesting thing which came out of that was, first of all, because the Maize genome had just been sequenced, was we got 129,000 novel splice junctions. So they suggest there are new genes and new alternative splicing products out there. Alternative splicing has turned out to be one of the sort of clues in which there are very quick responses of the genome to changes which are taking place, so changes in the environmental response. So those were good clues to follow. In addition, we found out a number of intergenic transcripts, 7,300. We blasted them to rice and other genomes, and we found out many of them actually hit rice genes, so those are novel genes, and many of them were drought expressed. The differentially expressed genes, essentially, if you look at it in certain Q values, 0.005 to 0.05, we get a different number. It goes all the way up to 3,300 alternative splice drought expressed or differentially expressed drought genes. So that's another level, essentially, of regulation out there. Then we did the normal functional annotations, look at geo categories, and we had 3 different tissues: vegetative, reproductive, and reproductive. Vegetative means this is vegetative stages. At the same time, we harvested the reproductive tissue. And we found many of the processes which are downregulated, for example, photosynthesis, sugar metabolism, etc. Didn't tell us very much because annotations were not good, so I'll come back to that in a minute, how we got the better of it. But we also found that the differentially expressed genes in terms of the alternative spliced isoforms. There were a large number of them involved in photosynthesis, cell division, translation, etc. So there's a clue out there to some sort of short-lived transcripts which could go through some sort of decay and very quick regulation in response to the environment. When we put all these genes into MapMan, which is essentially a method in which you can look at all the biochemical processes out there, what you vastly see out here, there's a whole lot of red across many of these pathways. Red is essentially downregulation. Downregulation essentially in lipid biosynthesis, cell wall biosynthesis, secondary metabolism, amino acids, etc. Some essentially out here are increased. Most of them are decreased. So when you look at them in detail, especially in terms of what we were looking at, this is the ovary tissue of Maize, which is the most sensitive stage. If you have stress out there, you have a few percentage of your kernels only there. So looking at these, we in both in terms of starch and sucrose metabolism, here we have a number of genes which are plus, and in this case, this set of genes 11, I'll come back to. This is the annotation of all those genes. These ones are all the invertases. And so we saw a number of invertases. We did quantitative RT-PCR and see most of them here downregulation shown here. This is leaf meristem tissue and ovary tissue. So in ovary tissue, all of them are downregulated. In the leaf meristem, in the same plant where we harvested the reproductive tissue, these ones, some of them were upregulated, suggesting there probably is a turnover from the leaf to the ovary. But anyway, genes like, for example, cell wall invertase 1 and 2 were downregulated, which then gives us an idea that these are potentially some of the regulatory steps we would like to address out here. This is as far as we are getting to in Maize right now, because what we are doing is we're looking at most of these genes in terms of an association mapping population, which is developed by the Generation Challenge program, and we are looking at some of these out in that population. But just looking at some few processes which hadn't been identified before, this is one step, essentially, the number of genes involved in cell cycle. So many genes involve cell cycle, G2, G1 phase. In the G2 arrest, G1 arrest, you can see many of the genes are minus, all negative, all repressed, except for a set out here in the G1 arrest. So cell cycle essentially is vastly down-regulated a process. And in addition to cell death, probably this is one which slows down most of the development of the ovary tissue. So there's down-regulation of cell cycle genes. Now we switch then to rice, because we have now models essentially in maize, which we expect now we can get certain phenotypes based on natural populations. But rice is a very good system to identify gene functions at a mutant level. And so we developed the same system in rice. This is an example of drought stress response in seedling, vegetative tissue, reproductive tissue, number of genes, normal Venn diagrams, upregulated genes. I normally show that in blue, and reddish tinge as downregulated genes. Okay, there's a large number of genes. What do they mean? If you look at gene ontology categories, you get upregulated genes, downregulated genes, and we compare them all. I'll come to those in detail. They themselves don't tell very much unless we go down to Go categories which make some biological sense. First of all, what we did was we looked at in these classes what were the overrepresented cis elements in certain classes. So cis elements which we found by de novo discovery, a number of new ones which were in different stress, either in vegetative, reproductive, or both. And these, therefore, then later on come to be useful when we identify transcription factors involved in regulation of a particular process there. So with all that raw data, what we wanted to do was go from these genes. We don't want to study genes by themselves. We want to look at it in terms of modules, and the modules in terms of networks. And the reason being that the idea of co-expression networks is that essentially genes which have a similar expression pattern have got some sort of brotherhood, some sort of reason to be expressed together. So they may or may not be completely differentially expressed in the tissue, but even if they are present in that tissue, they have some sort of function together. So they're most likely involved in, to be functionally associated. So we developed a rice environmental co-expression network, which we call RECON. We took all the publicly available data sets for rice, all which were involved in some sort of environmental response using Affymetrix arrays. So Affymetrix arrays, there were 129 of them in number of groups, number of experiments, and we took the data from all of these. We re-annotated these Affymetrix arrays because they were not using the uniqueness of the probes. We used only unique probe annotations, so we could get a large number of genes from the Affymetrix arrays into our data set. Then we did a correlation, use correlations to get a co-expression network. The first networks were about 34,000 genes with 18.5 million edges. Depending then on the cutoff we have for correlation coefficients with them, you can have many of them together, or you can disperse them into more dense networks. So we took dense clusters in which, with a certain cutoff of about 0.67, so we got about 28,000 genes, 20,000 genes in 1,700 clusters, which are more dense. Therefore, these 1,700 clusters are probably having more of a functional similarity together. And this is what we based most of our studies on. We then took our drought expression data, superimposed it on these clusters, and found out which clusters then out of these, which were originally obtained from environmental stress experiments, which of these are drought responsive. So our workflow essentially was two ways. One was we made a network of these, shown in this part, and then we took our own data in drought response, and we mined clusters in different stages and find out genes which are biological processes which are similar in all of them. We then pointed out to certain candidate genes which seem to be important in sort of regulating or driving such clusters. And we started to look at mutant resources in rice, which are quite a few. There's about 200,000 essentially which are available, knockout lines, which are insertion mutants. So we pulled out about 50 transposon inserts, 200 tDNA inserts, knockout lines, and for those genes which didn't have essential genes, which we thought, which didn't have any inserts in them, we did RNAi downregulation. So at present, we have already developed about 50 homozygous knockout lines, which at present being phenotyped. I won't tell you the results of these ones, but many of these we have been looking at in terms of overexpression analysis, and that's what I'll describe today. So we hope then finally to get both a knockout study and overexpression study based on them. So in any of these drought modules, this is one drought module in which you have number of genes shown in either circles or triangles. Triangles are transcription factors, which are or regulator genes, which are driving the network. Blue shows they are all expressed or overexpressed, or they are induced under drought, and they're induced under drought in three drought stages. So this particular cluster has got a large number of drought-induced genes, which are both transcription factors, other regulators, including, for example, kinases, phosphatases, and other regulators. Interesting thing about this cluster, we have then 4 drought-tolerant genes. This has been identified by other studies in rice. In rice, there are about approximately 150 genes whose phenotypes are known to be involved in drought response. So here's 4 of them in this cluster, which show some drought response, altered drought response phenotype. So this validates essentially such a cluster is probably involved in a function in terms of drought tolerance, and it'll be interesting to see other members. What's clear then, actually, there are many members which are not regulated. So there's white, so they're not blue, they're not upregulated. So many transcription factors also, for example, here there's a NAM transcription factor, which then could be involved essentially in the process, but we wouldn't identify it by just induction under drought alone. This is a cluster in which we have both upregulated genes and downregulated genes. And in this network, essentially, we have genes which are essentially looking at upregulated under drought in the reproductive stage, and linked to genes which are downregulated. So there's a set of genes which are downregulated, and a few genes here, and this is upregulated. And these are linking to invertases. This is a rice cell wall invertase, has been shown previously to be involved very specifically in induction of drought responses. There's another invertase here, vacuolar invertase. So this is one of the reasons why we choose this cluster and search for mutants and overexpression phenotypes of these genes here. In summary, these are a number of different clusters which we have. The clusters, each of them having a large number of genes. You have 71, for example, here, 1,700 genes. Each of them have drought tolerance genes in them. So suggest by guilt by association that some more others in that cluster might also be involved in drought tolerance. We just got to get phenotypes for those. Here you can see some of them which are known genes. Many of you who know them just can read them out. NAC transcription factors, then a DREB2A, etc. So we are looking at these genes with knockout mutant phenotypes and some overexpression studies. What we also found out, if we try to integrate this data from responsiveness, essentially, of genes here. So this is co-expression clusters. Here, for example, the first column is a drought reproductive tissue experiment. These are all repressed under drought here and upregulated under reproductive drought. And there are other clusters which are involved in development, etc. If you look at any particular class, for example, there's a cluster out here, and look at them across here, what we see here is what we have calculated here is the responsiveness of genes. So responsive genes are those genes which essentially change very much under certain experiments. So many of these drought responsive genes are changing under many development stresses also. So drought responsive genes also responsive to many other stresses. And we see that it is reflected in terms of the histone and DNA methylation. So DNA methylation is increased or decreased, and histone methylation increased. So responses of genes is also involved somehow or the other in chromatin changes. Then we looked at if these clusters, which we see essentially from gene expression studies, how do they look at across the genomes? So we took about 78 QTL positions in the genome, and we asked for any QTL position, like this, for example, would be one particular QTL position. How many genes of this cluster? For example, in this cluster, there could be 100 genes out here. How many of these genes are present in that particular QTL cluster? And for example, is it enriched for them? So if we see that, for example, for many of these QTLs, in terms of the red, we see an enrichment essentially of genes which essentially are expressed under drought, and therefore are candidates essentially for that QTL, either in being the targets for the gene itself or involved in the regulation, co-regulation of the QTLs. This is just one sort of way we look at in terms of a broader aspect of gene regulation. Now I come to the phenotype which we've been interested in, and especially because of studies by John Pasiora, we've been studying essentially in terms of water use efficiency, because it gives us a model in which to study yield. Yield in terms of a function of water use efficiency, transpiration, and harvest index. So a few years ago, we had identified a Rhabdopsis gene called Hardy, which overexpression in rice showed us higher biomass and increased water use efficiency. So using such a model, we looked at now genes which essentially would give us a higher biomass and water use efficiency, and then look at yield changes. And most of you are aware of all the terms involved in this. I won't look at that. So I'll now deal with one sort of gene which we use then to dissect the process, water use efficiency, and yield. So this particular gene, a transcription factor, which is induced in the seedlings, vegetative tissue, pre-anthesis and post-anthesis. It's a rice higher gene, which we identified from the transcriptome data. It is a reproductive tissue in the P3 stage. It's expressed. And so it's consistently across different tissues it's expressed. So we did a simple 35S higher overexpression construct transformed into rice, Nippon barley, and 3 other cultivars. Get these transformants, fix them as homozygotes, and then conducted a drought test in which at day 50, we stop watering for a while and then give a constant drought stress, about 70% field capacity for about 20 days. So 20 days stress is given, and then we see the change in growth. We compare then these higher lines compared to the control, and we see the change in growth. So the results are here. Here is the wild type versus number of higher lines. The shoot biomass under well-watered conditions is higher, significantly higher, and also under drought stress conditions. The water use efficiency is also higher, increased in both in drought and well-watered conditions. Photosynthesis, as seen as under LICO measurements, is also increased in these under well-watered and drought stress conditions. And then the first result of photosynthesis essentially, for example, sugars. We looked at sugars. This is total sugars that is increased in the well-watered and the drought stress conditions. This is also represented also in sucrose, glucose, fructose. All 3 are increased in these cases. Therefore, we have a case in which we have increased biomass, increase in photosynthesis, and the primary products in which we have under sugars too. In the drought stress test, the first test we did was progressive drought. So wild type versus 3 higher lines. So as we progress drought, that means you just cut off water. As we progress on up to day 6, you see the wild type well, dies off or wilts off, and the higher lines survive, or what you see out here is the relative water content of the transgenic higher expressing lines all out here versus the wild type. The RWC reduces where these ones are maintained. So these plants survive, and then we also study yield data on that. But what we also saw is during both drought and the well-watered conditions, the photosynthesis of these lines, higher lines, in terms of well-watered conditions and drought stress was all increased. So photosynthesis was maintained with these higher lines under drought stress. The phenotypes of these lines seems quite interesting. What you see quite simple to see out here. The higher lines have increased photosynthetic pigment, much more greener. If you do analysis of the pigments, of course, they also are increased. The chloroplast number is increased. This is wild type versus the higher lines in large number of staining chloroplasts. And if you look at the plastids, essentially of wild type under drought and higher under drought, the wild type a bit more collapsed under drought, whereas the higher lines have the plastids a bit more resistant and therefore probably photosynthesize for a longer time. So a large number of processes involved in photosynthesis are maintained under drought. This is a cross-section of a TEM image of a flag leaf, and what you see then is wild type versus higher, and you see white starch granules. The number of starch granules are increased out here compared to the wild type. So even another step in photosynthesis, and the primary product has been increased. When we take these lines and subject them to increased light intensity, so increased light intensity, the assimilation rate essentially of these lines increase while the wild type sort of levels out. So there's more capacity essentially in these genotypes to photosynthesize. So we did the normal experiments of doing microarray studies in these higher producing lines. And what we then got was, okay, a set of genes which are upregulated, downregulated. And what you see out here is just a short comparison of genes involved in photosynthesis and carbohydrate metabolism, which it shows you the correlation, or well, the correspondence in these cases. And both cases, there is a concomitant increase in these photosynthetic genes and carbohydrate biosynthesis genes. So we could then use our data of microarrays to build up a hypothesis on a broader scale. We also had an interesting root phenotype, not unexpected. This is the higher lines at early germination and increased adventitious roots, and as number of lines having a large number of thicker roots here. Cross-section of the roots show you a large, thicker cortical cells, which are arranchymus cells, and probably then Amelia will tell us exactly what the functions would be. These are all certain drought responses, which are then positive to give you a drought-resistant genotype. The biomass of roots has also increased out here, both on the well-watered and drought conditions. We then took plants which had been stressed at the reproductive stage under drought and also well-watered conditions, and compared the yield of these plants. So what you see out here are different parameters of yield: spikelet number, plant biomass, grain number. This is all greenhouse data. Okay, grain number, grain yield, single grain weight, and many of these are significant in terms of the higher lines under drought stress, and under in this case, sorry, this is only under this is wild type and higher lines under normal conditions. Under normal conditions, there's higher number of there's increased biomass, grain number, grain yield, etc. Under drought stress, show these spider diagrams. It's more representative. Under well-watered conditions and reproductive stress, many of these components, same ones, have increased in all these higher lines compared to the wild type and reproductive drought and well-watered conditions. So many of these parameters in drought stress response have increased in the higher lines. Now, if you sequentially look across them, it starts with photosynthesis, a number of genes involved in carbohydrate metabolism. And what's interesting, the way I see it, is that if you have an increase in sugars, you normally would expect a downregulation of well, many of the genes involved in in photosynthesis, so sort of a sugar sensing. So we don't see a feedback inhibition of sugar sensing. Either that's because the sink pulls out enough of the sugars, or that it has become sort of more neutral to the amount of sugars present out there. Now, to understand all this was just the phenotype. To understand all this and to make something out of it, we looked at essentially what is happening in these genotypes. So this particular gene, higher, is a transcription factor, AP2 transcription factor. We identified it from this sort of analysis, in which we looked at, so AP2 transcription factors are well known because of their response to drought, and some of them have been shown by overexpression analysis, at least, to be involved in drought resistance. So we took a set of 35 AP2 transcription factors of rice, and we looked at it in terms of association of these transcription factors to biological processes which we had seen downregulated under drought. So these are the downregulated genes, processes under drought, all in terms of GO categories of biological processes. For example, this is starch biosynthesis process, glycogen glycolysis, etc. So if we look at associations are shown in this color code here. A positive association, which is a transcription factor GO term, it is Z-score up to 3. So these blue show high association of your transcription factor regulation and the regulation of these particular GO categories. So here there's a high level association out there, and here in yellow are negative associations. So when you compare all these transcription factors, but these are all drought regulated AP2 transcription factors, we find out that there's only one, and that's this particular one, which is higher, which is completely positively associated with all these particular processes. So higher is the only drought-induced AP2 transcription factor that has positive correlations with all these biological processes. So this gave us another clue in which we search for associations, essentially, of transcription factors to biological processes which are essentially sensitive to drought. So this is what we are following in other overexpression and knockout studies. Now, higher association with these GO categories or GO terms under no stress and under stress conditions were then studied using this sort of a study in which we look at the environmental conditions. So environmental conditions, for example, these negative conditions, which are stress conditions, and then developmental stages. So these all are, each of these points are GO categories. That means biological processes defined under a certain term, let's say photosynthesis, a specific process, etc. And those terms, how they are related in association with environmental conditions, positively, negatively, and with certain development stages. So here you see the Z-scores of them. And what we see, a large number of these processes which are positively associated with development stages required and activated in these stages, like, for example, during photosynthesis, during reproductive stages, etc. These are normally downregulated under environmental conditions, as you've seen earlier. And if you look at what these processes are shown out here, so these are all downregulated under drought or different environmental conditions, and regulated on development conditions, cellulose biosynthesis, glycolysis, glucose catabolism, etc. So these are the type of processes which we expect, which are downregulated under drought, which in these cases has been maintained under by higher expression. So this is one of the reasons why higher gene activity can maintain drought stress resistance. So we come then to certain concluding looks at what the higher gene expression tells us. So higher expression in rice when higher is expressed in rice, a number of genes are upregulated, and those genes are involved in several traits involved in abiotic stress, growth, and yield. These all stand for a number of genes and biological processes which are involved in root dry weight, drought tolerance, plant survival. So higher essentially is able to upregulate these genes. It's also involved in upregulation of genes involved in root branching and stomatal frequency anatomy processes, and also involved in biochemical processes like chlorophyll ratio and alpha amylase activity. So this is the way we look at a bit more broader processes which this particular gene regulates all these steps, not just the genes per se. Then when we look at higher response in rice to see what are the processes which are regulated, higher has positive associations essentially, which we've seen before, with carbohydrate biosynthesis, photosynthesis, and chlorophyll biosynthesis. So in our model, these processes are upregulated upon higher expression in rice. Higher upregulates these processes. But these associations are disrupted under environmental stress. So these processes which are also downregulated by drought stress in rice. So stress essentially is very severely deregulates or downregulates these processes. On the other hand, higher is induced by drought. So drought stress essentially induces it. So here we have this cycle in which stress can induce higher, higher can induce these processes. But stress essentially is very strong, which essentially downregulates these processes. So our model essentially then comes down to, in this process of stress higher and these particular processes, under well-watered unstressed conditions and short drought stress, higher can activate these processes. But under continuous drought stress, although higher is induced, but by some other pathways, there's very strong downregulation of these processes. And so essentially what happens is there is downregulation under severe drought. But when you increase the expression of higher, then it's able to compensate essentially for that stress, and it keeps these or maintains these particular processes. So that essentially is our model for expression of higher. So we're testing this in different systems, including different plants. The first which we started was with different cultivars of rice. So this is comparison of higher in the normal cultivar Nippon Bare, 3 days of drought compared to wild type. In IR64 at 7 days, higher lines and wild type stressed. Cocodri, which is a US cultivar. So this is under 7 days, the higher lines compared to the wild type stressed. And drought resistant cultivar, Vandana, which is also showing you much more survival. At present, these are only vegetative tissue ones. So these plants have been recovered, and we're looking at drought yield under drought for these plants. So this is the way we have been looking at how you can use certain genes, transient factors, as probes in a whole network, identify processes out there, and therefore probably get to processes which you would like to have either regulation of it in transgenic systems or use them as functional markers in some way. Most of this work which I spoke about is acknowledged to be from work I started over at Virginia Tech at Virginia Bioinformatics Institute. Bioinformatics student Arjun Krishnan, who then continued to be a postdoc, then Batlang and Madan both worked on transgene studies on that. The Maize work was done along with Ruth Green and a graduate student of hers at Virginia Tech. At present at Arkansas, we're studying other phenotypes of this, including protein networks involved with higher expression. So to find out what are the cascade essentially of genes involved in it. At Penn State, protein interaction networks are being studied by Yunong Yang. And at LSU, Niranjan Bayasak did some of the transformers which I showed you out there for the difficult genotypes of rice. With that, I'll leave you and open to questions. Thank you.